Surreal.
Okay, we are live. We are recording. Phone lines are open. You are good to go.
Welcome to the trustee meeting tonight. There's not quite as many of us, but we'll get it done. Could everybody stand for the pledge, please?
Pledge allegiance to the flag of the United States of America and to the republic for which it stands, one nation, under God, indivisible, with liberty and justice for all.
That was pretty good. Could you call roll, please?
Certainly. John Holter,
Here.
Francis Bach, David Cataletta,
Here.
Tim Garnell,
Here.
Ben Dollinger, Jim Grimes, Bill Taylor,
Here.
Susan McGraw-Keever,
Here,
and Mike Martinson. Okay, thank you. We have 4 absent. We have a quorum.
We have a quorum, just, but, is there anybody on the line? Anybody calling in?
There's 0 callers as of right now. Thank you.
I think we should go right with Dr. Goldberg.
Okay,
Welcome, doctor. Welcome.
this is our annual report from Goldberg Labs at Sunnybrook University. Thank you for coming.
my pleasure. Happy to be here. I have a lot of results to get through here, so let's just dive right into it. Obviously, you're welcome to ask questions as I go, although there's a lot here. So, and you can also save your questions for the end.
Would you have a preference?
I think if there's a question as I'm going, it may be good. I might be able to quickly clarify. Yeah. So you're welcome to interrupt. And hopefully I'm going to move kind of rapidly, but I'll slow down at any point. This is your presentation. So, and as you know, I sent this report out on Friday, a little bit later than I wanted to, but it got there. And obviously, follow up can be done at any time and in any way. So we can have follow up meetings, talk about what the results mean, implications, future studies, etc.
I'm sure that we will want to do that at our water quality
Yeah,
committee.
we're gonna
have to have a meeting and discuss everything.
next year.
Sure thing.
Yep. Sooner. How's your
Yes.
schedule looking?
After this week or so, it should be pretty good. So
Okay.
yeah.
Terrific. I'll be in touch.
Great. Okay. And yeah, this is the 9th year of data. So we're actually going into this coming year will be our 10th year of sampling. So able to pick up trends, with it. So let's here. Okay. So, we all know why we're here. You're here. Your jobs are protecting the resources here, and there's incredible resources, water resources all across the town of East Hampton, of course. And that goes just from enjoying it recreationally to the incredible fisheries here on the East End. Everybody knows the renowned fisheries, be it shellfish or finfish, of the town of East Hampton. And of course, in many ways, the town of East Hampton is set up to have the best water quality in all of Long Island. It's surrounded by the ocean and Block Island Sound and Gardiner's Bay, and all these water bodies, exchange very strongly tidally, and therefore should be keeping all of our waters clean. But we also know that there's been a significant changes in land use over the decades in the town of East Hampton, and there are some areas that are quite built out compared to some other areas. And we know we've learned the lesson, of course, across all of Suffolk County, how that can influence land use like that can influence water quality. I'm just general primer. I'm going to talk a lot about algae today, and for good reason. On the one hand, you can't even see them without a microscope, so one might think, why bother? But on the other hand, it's just a known scientific fact. It's the algae at the base of the food web that supply the energy for all the rest of the food web. And so all fisheries, I teach this in my biological oceanography class, you can't even begin to model a fishery until you know what the rates of primary production are by the phytoplankton. They produce half of the oxygen that we breathe, and so are vitally important. And this is just showing how important they are for fisheries. I guess I had the slide already queued up for that. We will talk about some harmful algal blooms. These are some algae that can grow to levels that can be of concern with either the ecosystem or with regards to human health. And I'll detail those as I go. And again, on the effect on fisheries, of course, the recent severe collapse in the bay scallop fishery is obviously large in the minds of many people, and I'll have a little bit to say about that and what our findings are and how that, with regards to what's happening to that fishery presently. And I'll be talking about pathogenic bacteria, also known as indicator bacteria. These are bacteria that can move from land into the water with implications for closing shellfish beds. So the New York State DEC declares every bay bottom across all of New York State as either open, closed, or temporarily open to shellfishing. And they use fecal coliform bacteria as a measurement there. But there are other important types of bacteria to measure. For example, the Department of Health measures Enterococcus. And also, the source of these bacteria are generally unknown. The DEC just measures them and then says, yes, good, or no, don't shellfish there. So that's why we use, and I'll talk more about our microbial source tracking approach, where we can figure out the precise types of bacteria that are causing these systems to be opened or closed. So overall, these are our objectives, and basically we're looking at water quality from top to bottom, and how it might affect shellfishing, swimming, and overall ecosystem health. And we're in marine waters as well as fresh waters. We sampled a record 23 different locations across the town on a regular basis last season. And that generated really an unprecedented data set as far as the amount of data that we have. A lot of our measurements are made right on the spot. And in some cases, we have devices that are in the water making consistent measurements with us not needing to be there. And then in other cases, we're bringing the samples back to our lab in Southampton and making the measurements there. In many cases, we're using, in most cases, state-of-the-art methods. And I'll point out where we have certifications from, for example, the state for our methods. And we always try to interpret our findings in light of whatever state and federal standards are. So if we make a measurement, what does that mean? Well, there are in many cases, there are guidelines to tell us exactly what it means. So here's a map showing all the locations that we sampled in 2021, and you can see it's rather extensive. Many locations throughout, and you can see we went as far east as Fort Pond, as far west as Wainscott, and north West Creek. In some cases, some of our sampling was done in partnership. For example, the Concerned Citizens of Montauk got us the samples from Fort Pond. But in almost all the other cases, it's the Global Lab out there collecting the samples and again, bringing them back for analyses. And just for record, and it's in the report, if you're ever wondering where a particular site is, this table in the report indicates the exact longitude and latitude, as you can see, down to the umpteenth decimal, so you can really track these locations on your own as well. And like I said, more sample locations this past year than ever before, and done purposely because we had some very specific questions that we're seeking to resolve. And I think our collaborative effort to choose where we'd sample and when did its job, and that we were able to get the answer to very highly specific questions that we had, at least for some systems. Okay, so with that as background, I'm just going to jump into what we found and start with something that's kind of boring but important at the same time, and that's temperature. And actually very interesting, 2021 was actually a cooler year compared to years prior. We kept seeing very, very warm summers. Last summer was not as warm as some previous ones. You can see not a huge variation amongst the sites either, but again, slightly cooler, which bodes well for marine resources, particularly those that we think are at some of their extremes in thermal tolerance. And this is a foreshadow to say, for example, the bay scallop. Well, I'll talk more about it later, but that would be one of those organisms. And also in good news, the total levels of algae in the water were just about in a good area for most of the year. We had a couple of excursions, as you can see here, that I think were both in Hog Creek and in 3 Mile Harbor. But by and large, what you want to see is somewhere in the zone of around 5 or so, and that's basically what we saw. As you can see, some places it gets higher. EPA and NOAA both concur that once you get above 20 micrograms per liter, that's way too much, and that can have ecosystem impacts. So only a few samples hitting that criteria there. And this is just past data just showing that in East Hampton waters, the amount of algae you get, the amount of phytoplankton, directly correlates with the amount of nitrogen loading from land to sea, both the total amount and specifically the amount from wastewater. With 3 Mile Harbor traditionally having the highest nitrogen loads and also the highest levels of chlorophyll. To get into some of the harmful algal bloom species that we're monitoring for, the first one I'll talk about is Alexandrium, probably the most dangerous from a human health perspective. It makes that compound in the upper right known as saxitoxin. That's a neurotoxin, 1,000 times more potent than cyanide. It can get into shellfish, and when it does, it can lead to paralytic shellfish poisoning. So something important to monitor. And we did monitor that and see it around. You can see here's Accreditation Harbor, all 4 of the sites that we monitored, and all of them had Alexandrium. Thankfully, none of them at very, very high densities. This is typically a spring phenomenon, and that's why the dates are as they are. And you can see high levels in May and then sort of tailing off into June. That site 7B, I'm going to get it partly wrong, and John maybe will be able to help me, but that's Gerard Road, not far from the inlet there. And generally you'll see across the board had probably the best water, the best water quality in Accreditation Harbor. That'll come up again and again.
Culvert?
I think that site is right around where the oyster garden is
Oh, okay,
in Ackerman.
okay.
So a little bit further south from the culvert, I believe.
South of the
Yeah.
on the east side of the harbor.
Yes, that's right. So in 3 Mile Harbor, we had Alexandrium also show up at all the different sites. The lowest levels closer to the inlet, the highest levels in the back part. We did have one measurement here that was extremely high, over 2,000 cells per liter, and if that were sustained, that would be a problem, but thankfully that was just for 1 week. I will note, however, this is one of the types of harmful algal blooms that can change a lot year to year. So this is just showing the first, for example, 3 years of sampling in 3 Mile Harbor. But for perspective, the number we had last year is probably, again, one of the highest we've seen in 3 Mile Harbor. But again, thankfully, no major issue there.
Chris, were there other areas in the island that had high counts of Alexandria?
So no, last year was, and it changes year to year, and it really depends in large part on how quickly temperatures rise in the spring, and how long it sort of stays in the sweet spot. But last year there were no closures. The cells were around, but generally I think it was cool and then ramped up out of its sweet spot, at least sweet spot for that organism, too quickly for it. So by and large, it was a mild. There were no closures in New York last year for PSB. Coccidemia is a persistent issue all across the East End. And I'm just doing a little bit of retrospective here because it's actually quite interesting. Way back, we would always see this originally in 3 Mile Harbor almost only. 2019, you can see the highest levels that we saw were actually in Northwest Creek. 2020, the issue was really in Accreditation Harbor and Hog Creek. And again, we're monitoring all these sites. And now in 2021, the levels in Accreditation Harbor were really low. And the high levels were persistent, sustained in 3 Mile Harbor. So this is an organism that it's probably the trickiest of them all to figure out, in that they just showed you 3 years of data where the peak, the peak of the population in East Hampton waters has been shifting and shifts amongst the different locations. And if I just go back to this aerial, because I'm not sure I'm going to have it elsewhere, you could just see this organism in general is quite patchy. And you can be in one area where the water is extremely dense, and then just go, not far from there, and it's fairly clear. And clearly it's doing that amongst ecosystems as well. And so, but this is, as I mentioned before, of great concern. This is an ichthyotoxic organism. This is an old publication from about a dozen years ago or so here, but just showing that it is toxic to bay scallops. A bay scallop won't survive that long if it's consistently exposed to one of these blooms. And unlike the Alexandrium, the rust tide was very widespread throughout the Peconic Estuary last year and Shinnecock Bay last year. That's the second year in a row that happened and persisted a long time. If you look at those two dates there, and that's not even the beginning and end, but just showing two incredibly dense periods and locations. And so the Peconic Estuary was really thick with rust tide for most of the month of September and the beginning of August as well. And so there are links here. I don't have a chance to go into all the details. I am not going to say that the rust tide is causing the collapse in the scallops. That is not the case, but it's definitely a contributor. We also know that temperature is an issue for the bay scallops. Interestingly enough, if you didn't know, on the one hand, bay scallops are found from Canada to the Gulf of Mexico. But on the other hand, that's the species. The subspecies that we have here, Argopecten irradians, this is really its southern extent. And so what you really have then, and if you think of an organism at its southern extent, that means it's at its thermal maximum. And so, you know, how long it's able, this subspecies can persist in our waters is an open question. But as a reminder, you may recall the Long Island Sound lobster was an incredible fishery in New York, and it just got too warm. And so I'm not saying that it's all temperature either, but we do know that it's close to its thermal threshold. And then I'll also say that when you have warm temperatures, by laws of physics, that leads to low levels of oxygen. So we have a lot of things happening all at once: rust tide, high temperatures, low oxygen. And it looks like these multiple stressors are all putting this fishery in a compromised state. Add to it that it's also known that it has a parasite, you know, it's all these things all together. We know it's not just a parasite because you have similar levels of infection, for example, in Nappy that you have, say, in Flanders Bay, but yet all of them die in Flanders Bay, and they're living and making it in Nappy. So there's certainly more than just that, but when you add in all these things together, it ends up being a problem.
Just as a bit of an aside about the Cocodinium, I saw the high numbers in the head of the harbor at 3 Mile Harbor, and I asked, our shellfish hatchery has a flupsy system down there, growing clams. And I asked Barley, you know, if he saw any impacts, and he said that they're getting very good growth, clams down there.
Yeah, that's great.
I'm not sure what the impact on clams is. I know it's not, you know, the impact that
they're probably the most hardy of we actually have a paper we published just recently where we did side by side the scallops, oysters, clams. The clams are the most resistant, partly because by the time they get to that time of the year, they're a little bit older and they literally just close down. So for the time when they're exposed at a high level, they're not really feeding. But in a similar sort of situation in the Southampton Marine Lab, we had at least twice in the last 5 years, tens of thousands of oysters die with the waves of this stuff coming through, and scallops as well. With the scallops, I think there was actually for both the scallops and definitely for the scallops, no survival, 100% mortality. And for the oysters, it wasn't full mortality, but again, this is also size dependent, right? So it depends on the size of the oysters that you have as well, and the clams. Although less so for the scallops. The scallops, actually, it was both first year and second year. So they're the most sensitive of them all, partly because of their very high metabolism that's related to their very fast growth rates. Okay, so transitioning now to other water quality parameters and oxygen. Here are the levels of oxygen that we measured on average during the day at all different locations, also showing the minimum. And so in general, things look very good. But of course, what we've come to know in the last decade or so is that oxygen can change a lot from day to night, and that relying on daytime oxygen measurements is probably not the best approach, partly because during the day, you will always see high oxygen because there's lots of photosynthesis. And it's at night that the levels drop down. And that's why continual monitoring is so important. it gives you sort of, in some ways, if you look at this isn't the cleanest data. It may look messy, but this is the way the oxygen actually looks, with each day-night cycle having a high and low period. And so what you see in that back part of the 3 Mile Harbor, as you go through summer and you enter July and into August, you see nightly excursions down into low oxygen zones. Nothing anoxic, which is good, and nothing sustained for hours at a time, but certainly shows that this is an area that is the water quality could be better, and the clams might be growing even faster if they're, let's say, in a different location. We did the same thing in Nappy, although the Nappy data is a very fun story. If I was giving this talk, I think just 2 weeks ago, I'd have no data. I think John knows the story, but we put out a sensor array in Nappy Harbor to look at surface and bottom dissolved oxygen and temperature, and that array disappeared in the fall. And we, by the time it became winter, we assumed it was gone for good. But in happy news, we got a call just a few weeks ago from one of the Bay Constables that they were actually able to recover it, and we were actually able to download the data. And you know, some of the data here, you can see that with there's those straight lines in the bottom. We're not 100%. Those are at times when it just wasn't collecting data properly, and/or it was compromised. However, the surface looks great, and we feel confident in the areas away from that. But and the bottom line is this data is not different from what we've seen in the past, and that surprisingly, even Nappy Harbor does have sags and dissolved oxygen at night, which is not what we would expect to find there.
Was that probe found in the same location? Or did it get dragged somewhere else?
It got dragged somewhere else. Yeah. And I still, there's a telephone connection that needs to be made back to me. And that is, I think the Bay Constable then spoke to my field technician, who then went out and got it, retrieved it. But still need to get the full story. But again, happy to have these back. Another thing that we did new last year, I sort of talked about it as a proposed activity for 2021, and that is doing autonomous surface vehicle surveys. And let's see if this wants to work for us. There is a video here. Yeah, it will go, so I can play this and talk at the same time. There was a point in time due to supply chain issues, I was not sure this was going to come through. I was hoping to have it in July, and then I was hoping to have it, well, I was hoping to have it in the spring, and then July, August. I think we ended up getting it in September, finally. So what this is you can see it's a remote vehicle. Embedded within the vehicle is one of our exo sensors that can measure temperature, salinity, dissolved oxygen, pH, chlorophyll, levels of blue-green algae. We can even, we've even made, if you look at there's a bracket in the front there, if you can see it, we actually had that put on specifically for measuring levels of CO2. It also has a GPS device embedded within it. It also can actually do bottom soundings if you want to do bathymetry of a given water body. So all these things allow you to make surface measurements. And we've done this before, but this is actually because the GPS unit is integrated with the water quality measurements, and there's a whole software package that goes with it. It's given us sort of a, we don't want to look at that again. It's given us a whole new way of looking at things. So I'm going to show you a series of maps that we made. These were all done in the fall. I wanted to get these done in the summer, the peak algal blooms, but it was still, we had some great opportunities. So I'm going to show you a few from Accreditation Harbor here. Of course, again, this is fall, and so, you know, the trends you see in the fall would be different from the ones you might see in the summer, for example. And you can see here, there's different ways of presenting the data. I have alternative views that I thought people might like. Well, I wasn't sure what people like better. This shows the exact tracks, so showing the exact data. You can also do something called interpolation, where you can colorize the entire water body. But I kind of like this one because you can see where the vehicle went. Oh, and I should mention, so when you do these sorts of what we call missions for the HiCat, you can do them in two ways. They can be pre-programmed. That's what we did here. So this is hands-free. You in advance, you program how you want it to run. It runs that program. You have a telemetering antenna that's communicating back to it from a computer, so it knows exactly where to go. When as needed, you can overthrow the controls at any time. There's literally a a gaming joystick like you'd have on like an Xbox or a PlayStation. You can override the controls as needed. And if you want, you can do it all without any pre-programming and just do it all by hand.
Chris, how long did it take to actually do this particular map?
To make the map or doing the,
To actually have the vessel?
yeah, I think this was a few hours of effort. Yeah, it doesn't go super fast. It goes somewhere in the vicinity, I want to say, yeah, I think about 5 knots or so. Yeah. So just enough to beat the tides and beat a decent amount of wind as well. So, I mean, some of the trends you see here is that I think this was done right on the tide, so with the tide turning around. So what you can actually see is if you go near the inlet, you can see green as well as blue. So the tide actually turning and getting the cooler water coming. In this case, I think actually coming out. So the cooler being in the sub-basin and in the estuary in the fall because it's cooling down. And this was done in the morning. And I'm just going to show a series of these, but here's the salinity. In this case, you don't see the effect of warming, but you do get a sense of how the water is exchanging. So you can see that the saltier water closer to the inlet and the fresher water to the south there, and the very low levels of salinity to the southern southwest extreme, just showing you that that's an area that doesn't get flushed as well compared to the same distance to the north where the salinity is higher. So it looks like a little bit better flushing to the north than to the south in Accreditation Harbor. And, certainly this is something that can be explored further and not something I would have expected a priori, right? And that's one of the lessons of circulation patterns. You can guess what the circulation is, but you can never be 100% certain.
I had a couple of questions about this. I know this was a first year and sort of a trial.
Sure, yeah.
Stuff, but if this is, I would expect, and tell me what the reality is. I would expect that you'd see a difference in the colorations if you did this on a ebbing tide versus flooding.
Yep, depending on the system, but yes, exactly. Yep.
And I guess in the down the line, if this becomes sort of a regular thing, would it be important to sort of tie the cruises to the same tide so that you're getting,
I think
apples to apples comparison?
In general, I'd say yes, but I also say it depends what your question is, right? So if you want to, I think if you want to understand, if you want to be able to compare apples to apples 100%, right? If you wanted to see, say, a worst case scenario, you might purposely go on a low tide because it'll have very little, and for a system like here, very little of the water from the Pathogenic and all the water from Accreditation. If you wanted to see maximal, flushing rate, you might want to go on a high tide to compare how the whole system is when it's all filled up with water from the Pathogenic. But anyway.
The other question I had is on most of these tracks, there's an outside color designating the different salinity in this case, or temperature in the previous slide. But there's an interior green thin line.
Right. I think the green line is the actual track, and then it's sort of on either side of it is sort of giving more color to it, so you can see it better. Yeah. And in this case, dissolved oxygen again being a little bit higher by the inlet as we'd expect, and decreasing levels going away from the inlet, suggesting that there's actually oxygen consumption potentially in the system. And in this case, looking at chlorophyll, and you can see particularly the areas further away from the inlet having the higher levels of chlorophyll, particularly to the south and all the way going to the west as well. And this, I think, if we overlaid salinity, it would look quite similar, I think. This is 3 Mile Harbor, not doing the whole harbor. And in fact, had we had more time, particularly this summer, I'm going to try to see if we can't get some missions right into the head of the harbor or comparing that area, although that would take like sort of 2 different missions. But you can see in this case, the cooler water closer to the inlet. So I guess at this point in time, this might have been one of the first ones we did in the fall, and therefore It's still warmer within the harbor. You can see as you get to the northwest corner, that's the warmest water versus what's in the inlet, which is cooler. And kind of the reverse here with salinity. And based on oxygen, you see, actually the lowest oxygen along the shoreline there. And that could be for a host of different reasons, but suggesting oxygen consumption by metabolic activity along that shoreline. And the algae levels sort of increasing as you get further away from the inlet, which you'd expect. And then finally, Nappy Harbor, another area we did. In this case, you can see warmer water towards the inlet. So again, I know this was either the last one we did or close to the last one. So the Pathogenic water being warmer than it was actually back in Nappy. So cooler, and actually you can see by the actual temperatures. Now we're down to 15 to 18. So warmer from the Pathogenic water compared to what's in the back of Nappy. You see that a little bit with on salinity, but not as much as I would have expected. And then on oxygen, again, you're seeing oxygen consumption within the bay. But again, just really, you can see where that is, mainly in the middle points there. And no strong trend in chlorophyll. So these, so anyway, this is a first go around. I have the best of these I'm saved for last. I'm going to show Georgia Pond, but I think this is a useful tool. And actually as it turns out, all of our missions, we only got this, like I said, in September. All of our missions were done in East Hampton Town trustee waters. So you got the first out of the box. And to my knowledge, there's not another organization on Long Island that has such a vehicle. There's some that are subsurface that the university has that go offshore, gliders they call them. But as far as stuff that you can do in an estuary, I think this is, to my knowledge, one of a kind. And like I said, you got the first of it, and we're excited to do more with it here in the coming years.
Chris, if when we do this again for the coming summer season, when would we begin this process? How often is it done in each location?
Yeah, I think that's something we should discuss. I'm very interested in hitting. My goal was to have this in the summer last year, so we can see in those times when there is a bad rust tide, when the oxygen is getting low. To get a clearer spatial sense of what's going on. So I could see, for example, being able to run this in and out of 3 miles. So instead of the way we did it for 3 Mile Harbor this time was, by the inlet and seeing the whole harbor. What I'd like to do is do the inlet into the harbor proper, back into the head of the harbor. Or maybe that didn't come out right, but I think you know what I'm saying. To get a clearer sense, particularly with the rush tide, to get a sense that, how extensive really is this, as opposed to just having a couple of stations to look at. Because each time it's literally, each of these maps is literally thousands, if not tens of thousands of data points, because it's just making measurements every few minutes as it goes.
How far below the surface is this?
I think it's about a foot to 2 feet below the surface. So it's, and most of these systems are fairly well mixed, so it's kind of representative of what you see in the water column. Okay, so overall, I think we know this, but we talked about algal blooms, we talked about low oxygen. It's all being driven by nitrogen loading, and so in 2021, we looked at the actual concentrations of nitrogen once again. Which is, so just again, the root cause of everything that's going on here. So we have algal blooms, we have low oxygen, but up until 2020, we had no data on nitrogen. We made a survey of nitrogen measurements in 2020. We repeated and expanded that in 2021. It's worth mentioning, well, just go right into the data. And I've broken this up into system by system. So here's the first 3, Nappy Harbor, Hog Creek, and Northwest Creek. And what I've put in there is the 0.4 milligram per liter threshold that the Pathogenic estuary had for ideal conditions for seagrasses. So all of these systems look quite good. The back part of Hog Creek is the singular location here that had a couple of excursions there, but other ones are exactly where you'd want to see them. Moving to Accreditation Harbor, situation slightly different. You can see there are excursions for 3 of the 4 sites. Again, the one that's along Girard Drive was the best of the water quality with regards to total nitrogen. Higher levels and the most excursion back towards Shipyard Lane. But also some excursions in the other locations. Now, the other thing to note here, of course, lots of salt marshes in Accreditation Harbor as well. And salt marshes actually can export organic nitrogen. So that's, and I'll talk more about organic and inorganic momentarily. But overall, higher levels above that standard for a good part of Accreditation Harbor. And then in 3 Mile Harbor, the levels where you want them towards the north part, where the water's coming from the Peconic Estuary. Back in the head of the harbor, you can see some significant excursions there, well above, in some cases, double that threshold. And just one measurement above that when you go out to, it's at Hands Creek, I believe. Yeah. So, but many excursions, and we added in, Site 11A is head of the harbor, but to the south. And so, I mean, in a follow-up session, we can talk more about that, but I have this is giving me some ideas for some important follow-up in that general region. Essentially what it's saying, in almost every case, it's higher there compared to the harbor proper. So that gives you the idea that that's where the nitrogen's coming in from. So we're honing in on things here. We also measured inorganic nitrogen. This is not necessarily the best proxy. There's not standards for it. There is for total nitrogen. That's why we did that. But inorganic nitrogen also gives you a sense of, is there a lot of nitrogen coming in from a source in a given location? And I put 5 micromolar in there as that is not a state or federal standard. That's sort of my standard, that if I see a number above that, I know that there's somewhere a strong source of nitrogen. So for all these places, nothing to really be worried or concerned about. Accreditation Harbor, high levels, consistently high levels backed by the Shipyard Lane location. Not necessarily a surprise. Pussy Spawn is coming in from there, and there's a lot of, I think, within the watershed, some sources of nitrogen as well. And then once again, the Girard Lane site looking clean and pristine over and over again.
Why is your threshold slightly lower there? Or maybe it's a different reading.
Yeah, it should be 5 for all of them. So yeah. Oh, so it's a little off there. Thank you. Yeah, made a mistake there. That's it's off because I made a mistake.
No.
That should be somewhere. It should be slid up. It may be a mistake, or it may also well be the sliding of the slides around because the line was
It's a
made.
typo.
There's a number of them that would be
Yeah.
potentially just below the
Yeah. So overall, it's the Shipyard Lane that is the site that had those 2 large excursions. Good catch. Thank you. And then again, the 2 sites in the back of 3 Mile Harbor popping out as having very high levels. And again, that one very high level in October back there. I mean, there is a seasonal cycle here. You'll tend to see higher levels in the winter when metabolic rates are slower. But again, that gives me a sense of that there's more of the story there that I think needs to be followed up upon. Okay. Yeah.
Before we leave, Matt,
Sure.
can you elaborate a little bit on the difference between total nitrogen, total organic nitrogen, and dissolved inorganic nitrogen?
100%. It's worth reviewing that, and I was remiss to not do that. So total is what's in the water. So you get you're just collecting the water sample, you're digesting everything in there, even the living things, so the phytoplankton, the bacteria, everything that's in there. And oceanographers would tell you that has both particulate matter and what they call dissolved matter. Dissolved is what just goes through a filter, right? So that's total. And so you've got organic, inorganic, particulate, and dissolved nitrogen all together. And so even if you get, for example, a really high and dense algal bloom, the level, then most of the nitrogen's actually in the algae. That bloom dies away. The level might be exactly the same, but it might just transfer to the dissolved phase. So that's why it's a preferred measure, the total, because you don't get, you can get, for example, very low levels of inorganic nitrogen under an intense algal bloom because they've used it all up. So that's why total nitrogen is probably, of everything, is the best metric to look at. So total organic would just be taking this dissolved inorganic and subtracting that from the total. You can get dissolved inorganic by looking at the total dissolved nitrogen, subtracting out the inorganic nitrogen. So it's really the way you divide up the pieces of the pie, so to speak. But total is, as it infers, includes everything.
So dissolved inorganic nitrogen is telling you the most about what?
It's an indicator. In general, during summer, you really should see very little dissolved inorganic nitrogen because it should be getting taken up very rapidly. So when I see spikes in inorganic nitrogen during summer, it's a sign that there's a nearby strong source of nitrogen. So and that's why that was incorporated in this coming year, to pair that with the total, to just get a sense of is that, and that played out very well here. Again, when we look at the in 3 Mile Harbor, and specifically that last date where the level is just through the roof, suggesting that there's something that's delivering a lot of nitrogen to that source.
Okay.
Sure. Okay, transitioning now to bacterial contamination and fecal bacterium, which of course can come from many different sources. And I'll just emphasize, I've said it before, but just a reminder, there are many organizations that make measurements of fecal coliform bacteria. Our lab is certified through the New York State Department of Health through a program, an accreditation program they have called the Environmental Laboratory Accreditation Program. This is an incredibly onerous program to go through. The amount of record keeping and redoing samples and documentation is incredible. There's a lot of scrutiny, and we have to report back to the state quarterly. We get blind samples we have to analyze once a quarter. We have to keep careful records of the temperature of our refrigerators, the temperature of our incubators, the temperature of the lab, who's doing the analyses, when they're doing it, everything is recorded. So I actually have people in the lab who can't stand the fact that we're ELAB certified, because they enjoyed doing it the old fashioned way, where you start to sort of do it. But we think it's important. This generates legally defensible data. So this data would stand up in a court of law because all of the records are kept and they can be turned over to the court of law. And there's really, there's pictures of everything taken. So there's really no question with regards to the data. So I just point that out because there are many groups that make measurements of fecal coliform bacteria. And even if they follow the same protocol, in the end, they're not generating the same. The data is not equal because of like I said, this is legally defensible data. And so just as we focused on Accreditation Harbor and 3 Mile Harbor in 2021. And so again, here are the individual locations that we sampled on, and you can see again 7B, that site that keeps coming up so nice, sort of close to the inlet there. E5 is as well, but it clearly looks a little different, and that'll play out here for this data as well. As a reminder, Accreditation Harbor is a system that has all 3 types of bay bottom when it comes to shellfishing standards by the DEC: areas that are permanently closed, areas that are permanently open, and areas that are seasonally open/closed. Okay, so getting into some of the data for these different locations. Here you can see the standard for a single sample for shellfish is about 14 colony forming units per 100 milliliters. And what you can see is that in the back, again, that area by Shipyard Lane being consistently over that, well, that's what the DEC declares, so that's good. In general, much lower levels in the open area, that'd be 7B, although we did have one sample that was over in that particular case. But clearly high levels back in that Shipyard Lane region, and somewhat high levels in other locations as well. We also measured Enterococcus. Now, this is the standard the state health department uses for swimming. And so just another way to look at it, it's a different class of bacteria. It may or may not trend with fecal coliform bacteria. And you can see even the best locations had some overages on certain dates. But again, Shipyard Lane site being the most consistently over. And then site 5, which I believe is that's Louse Point. Also having a significant number that were above, or there's a couple that were above the standard.
Just real quick, what are your thoughts on what seems to be the June throughout? is spiked to that sort of through the roof, greater than 401 level.
Yeah, well, probably a rainfall
Okay.
that occurred, and it was a cooler summer, but I think it was a rainier than on average summer as well. So that may well be what drove that trend.
Yeah, because I noticed in some cases, the fecal coliform numbers and the Enterococcus numbers on the same day in some stations, not all of them, were very different.
Yeah.
Why I mean, I don't understand that.
Well, they're in the same class of bacteria, fecal coliform. But remember, it starts out with total coliform bacteria, and then there's fecal coliform bacteria, and then the Enterococcus is a subsample of that. These are also, of course, microbiological assays. So it's what bacteria are responding to a specific growth medium at a specific temperature. And they're different growth mediums. So it's just different bacteria reacting to it. Theoretically, in most cases, you would expect that if there's high levels of fecal, there should also be, or you expect high levels of Enterococcus, but not necessarily the reverse. Did I get that right? High levels, no, the other way around. If there's high levels of Enterococcus, there should be high levels of fecal coliform bacteria, but high levels of fecal coliform bacteria, who those fecal coliform bacteria are, they may not be Enterococcus. So they don't necessarily, they're subgroups within an overall group of the total coliform bacteria. In 3 Mile Harbor, we had 2 specific planned experiments that we performed. And that is, in the past, we saw high levels of fecal bacteria at the Gann Road site, which is E10, and at the head of the harbor site, which is EH11. And so both of those were at marinas. In both cases, we saw a higher human fecal signal than other stations. So we purposely chose sites that were paired, but further away to see if we saw an attenuation of the fecal bacterial, the human-derived fecal bacterial signal at these sites. Okay. So here you can see the sites in the head of the harbor having the most samples that were over the DEC standard for shellfishing. Somewhat similar for swimming, although there's a couple of dates, as you can see, that don't quite match up. That one in June, again, for the 2 locations, the Gann Road site and the site at Hans Creek. So we did, our plan was to do microbial source tracking. Susan, as I spoken to you, I don't even know when it was. Just like our HiCat was delayed, the same supplies that were used for measuring doing this microbial source tracking are the ones that get used for measuring COVID via digital PCR. So we actually had a scare that we were not even going to get this data in. We did get the chips in, and we did get the analyses done. And I think the data was extremely interesting. Just as a reminder, what this does is that there's a specific DNA sequence associated with deer, birds, humans, and dogs/small mammals that we can detect, and we use that to look at the relative abundance of different types of fecal bacteria in the water. And I've shown this data in the past, but just to give you a sense, this is the relationship between the amount of Enterococcus that we measure in the water column, and then using a microbiological assay, and then taking all of the different groups that we're able to measure via digital PCR and comparing those 2. And theoretically, what you'd want to see is that they're almost the same. In this case, the relationship's pretty good. This explains 71% of the data, which says there's probably some other bacteria out there we're not getting, but still, the fact that these are 2 different measurements, this is a pretty good correlation. Okay, so these are all the data we collected from all the sites in Accreditation Harbor and all the dates. For today, for the purposes of brevity, I'm going to focus on the averages, and that's what's shown here. Okay. Okay. So the trends here for me were quite interesting in that, I've told you, I guess, from the get-go, that the Girard Drive site continued to be and look extremely pristine this whole time. And remember, these are percentages of total, right? So they all have to add up to 100. So even if the levels are low, which they were there, you still have to add up to 100. And so that very clean site there, most of the bacteria that were present, there weren't a lot, were derived from birds. And what you can see is that percentage dropped off at the other sites, and we started seeing a bigger signal from small mammals and dogs, and then 2 locations with a human signal, Louse Point, and then also the Shipyard Lane location. Now, Louse Point also came up as elevated last year with a human, that is, also had a human signal last year. And note that there's a ship launch there. I don't think there's a marina, but there's at least a ship launch there. But it's the second year in a row where we saw a human signal pop up in this particular location. So it's not a huge amount, but the signal is there, and it was detectable 2 years in a row. So, and that will be a theme, and it's been a theme elsewhere also, that when you do see a human signal, there's not a lot of different sources. We know it's not, we're fairly certain in most circumstances, not coming from septic tanks, because the aquifer itself retains those bacteria. So it's more likely a direct source, and in this case, it may well be the boats. Again, not a huge factor, but it's contributing and we can detect it. And a little bit of the same signal at the Shipyard Lane site. So in that case, it's more curious, but in that case, it could be, could be a lot of different things. I won't speculate too much, but it's not a huge signal. But that site had the largest dog/small mammal signal, more than 50%. And again, having done enough of this, that signal seems to be very indicative of road runoff. So that is, you're getting either dog waste or other small mammal waste on roads and then being flushed into the system.
Chris, I have a question, and maybe I'm not understanding this as well as I should. The human aspect, you said, if there is, which is the darker orange color here, with all of the septic tanks that are on Girard Drive and all the way around the harbor, that's a very small percentage of things that are coming out of septic systems. Is that indicative of the septic?
So we don't think it's really the septics is driving that signal much. So the thing is about, if the septic system is, so long as it's not in water, and there is at least several hundred feet for the wastewater to travel through either the aquifer, so to speak, or the sand or whatever is there, those particles in the aquifer, the sand and the sediments and such, actually do a good job of retaining the bacteria, right? And this is, and so for example, if you live in a location where you've got your own private well, right? Well, the standards are your well needs to be at least 100 feet away from your septic system, right? And hopefully also that your well is up gradient, right? But your neighbors, and the reason that can work, and in most cases does work, because you do get the retention of those bacteria. So again, when we see the human signal here, in most cases, it's going to be from looks like boats. It's possible that you could get a signal from, if you had a lot of systems that were literally in water, due to, say, a sea level rise, that have now put a system that was formerly not in water. And and that's also why the health department, for example, requires, I think it's 4 feet between groundwater and wherever the bottom of a septic system is for that exact same reason.
We do have septics that are exposed on Girard Drive. There you see them.
Okay, well.
So it's kind of surprising to me.
No, I think there's way more septic systems that are involved that are in water than boats.
I agree.
Boats, you would.
But Girard Drive, there was no human signal. So, and so to me, that just says that between the combination of tidal flushing there, that again, that it's also showing that it's an area that was very well flushed in general, very good water quality there. So if there are systems that are directly leaching into the water, there's never a good place for that, but it's a good spot in that there's all signs are that site is flushing very, very well.
Okay.
I found it curious that both these sites and also the sites with a high human signal in 3 Mile Harbor are all sort of road end and some boating associated.
There's a lot of places there, where the depth to groundwater is really shallow.
Yeah.
Along Girard Drive, up by Shipyard, and through Springs, through a couple of those blocks there. You go down 6 feet, you're.
Yeah, I know Shipyard is a low-lying area as well. And again, that's when you have that combination of that low-lying area and the slower flushing rates in that area compared to an area like Girard Drive, that's going to be it more likely for that human signal to stick around and be measured, as opposed to just being whisked away on the tide, so to speak. Okay, we did this in 3 Mile as well. And again, I'm going to just focus on the averages. And as a reminder, we had the Gann Road and Squaw Road were the paired samples, as were Head of Harbor and Soaks Hide Road. And so if we look at those first two, again, the whole idea was there was a strong human signal in the year prior at Gann Road. And the idea of looking at Squaw Road was to see, well, does that signal dissipate away, or is it just that whole area in general? And so what we saw is that indeed it was a strong signal again. I think that's our biggest human signal of the year. And that indeed, when you move away from there further south, it's still, you can still measure it, but it's not significantly lower compared to what we saw at the Gann Road site. And instead, mostly at the Squaw Road site, that dog and small mammal signal, which again, in many ways is indicative of road runoff. And I'll just also note, if we toggle between these two, mostly birds here, and here, more of the dog and small mammal signal. And so one of the things that could be driving that is, I think, when you look at Accreditation Harbor, a lot of salt marshes there, a lot of salt marsh habitat, a lot of bird habitat, and therefore less direct road runoff, perhaps in contrast to 3 Mile Harbor.
And same thing with the difference between the head of the harbor and Soak Hides.
Exactly. So Soak Hides is very forested around there, very little going on. You see that again, just thank you for pointing out, John, the strong bird signal. So again, that giving you a sense it's more of the natural background population. And some of the deer signals showing up there. We only got the smallest of human signal this year in Head of the Harbor, which is good news, of course. But that made it to the point so we weren't able to make the same sort of experimental comparison between the Head of the Harbor site and the Soaks Hide site. But we did learn a lot by including the Soaks Hide site in general. So anyway, a lot of able to affirm the human signal associated with marinas, able to affirm the road runoff leading to a strong dog/small mammal signal, and the bird signal sort of being more indicative of just the natural background levels of bacteria.
Chris, did you say that deer might be included in the small mammal category? No,
the ruminant is the deer signal.
Ruminant.
Yeah. So and historically we don't see a strong signal from it's there, but it's not as strong as it's always small. It's never more than 10 or 20% usually. So,
okay. So overwhelming majority throughout the harbor.
Dog and small mammal. Yeah. Whereas again, and whereas it's more birds when you get to Accreditation. So.
And I've heard you guys talk about the road runoff over the years. Can you just elaborate on that? I kind of a hard time following that. So elaborate a little bit on what your thoughts on there. Is it just a dog? Is it other mammal? Animals just pooping in the road and on the grass and just running off. That's
basically that's exactly what we think is happening. Oops.
Raccoons, squirrels, fox.
Yeah. But every time we've seen a strong signal there, it's an area of strong runoff. And we've seen that in Georgia Pond and in Sag Harbor as well. Okay. So the last section is just the freshwater sampling that was done last year. And again, our sites are Fresh Pond. No, that's not Fresh Pond. I'm sorry. Fresh Pond. It shouldn't say Amagansett up there though. There's Fresh Pond that's here. Fort Pond, which is in Amagansett, Wainscott, Georgica, and Hook Pond, with 3 sites in Georgica Pond, 2 at Fort Pond, and others having just 1. Now, Fresh Pond, which is not, what town is Fresh Pond? Hampton. It is, okay, all right, sorry about that then.
Fresh Pond was dredged out
Yes,
last, the winter of 2020, 21.
it worked.
Yeah.
Because it's not a fresh pond anymore. It's definitely high salinity. So we can keep sampling it, but I think we might be moving it to our marine sites because it.
Well, it may close up again.
Okay. Well, certainly last year, all the sampling, all signs were that it was, you can see here, an average salinity of almost 25, which is, it's slightly lower than the other marine sites, but certainly not a freshwater site. And of course, Georgica Pond being a temporarily open system, you can see that differences, got up to high levels later, but on average quite low. I'll show that. Now, in bad news, there is a chlorophyll standard that the EPA sets for freshwater bodies. It's 8 micrograms per liter. All the systems are above that. So it's a more stringent standard than for the marine. The marine, they say 20, but for freshwater, it's 8. So they were all above that, and it kind of didn't matter whether you're looking at average or peaks. They all exceeded that standard. And I just throwing this in here because I wasn't sure how to put it. We did measure total nitrogen in fresh pond because we weren't sure where it would fit in. And now it's, again, you can decide whether it's marine or fresh, but it was often above, typically above that eelgrass stand for the Pathogenic estuary. This is just mean dissolved oxygen, doesn't really tell us too much. This is the chronic measurements of oxygen in Georgica Pond. You can see a few excursions below the hypoxic threshold, but not many during the summer. And in good news, with the opening of the cut in the fall, it never got anoxic, which is something we actually have seen in the past. Like literally, you may recall past years, 0 oxygen for 3 days in a row or so. Didn't happen, so that was good news.
This is a surface reading. That's a surface water reading.
It's just below the surface. That's the buoy that we have. So it's in the southeast corner and above that deep hole. And I think this was the last time's total nitrogen measurements. And I think that's the last year too. So I'm not sure why those are in here. But I'm just going to switch to sorry about those 2 slides. I'm not sure what they're there for. But of course, of the algae, even though all the lakes are above what we want for total algae, the ones that we're concerned with the most are the blue-green algae. And that's because they can make toxins, both gastrointestinal toxins and neurotoxins that can be harmful to humans, can lead to both, as per the CDC study, hundreds of illnesses of dogs and pets. We know, of course, historically, the dog death that occurred in Georgica Pond. Dogs have also gotten sick on Fort Pond, as well as Mecox Bay here in the South Fork. So this is the data for 2021 with regards to blue-green algae. So the easy things first, no blue-green algae blooms in Fresh Pond or Hook Pond to worry about. The other thing we can get the details on is that Georgica Pond had its most intense blue-green algae bloom since 2015. And we did have some exceedances in Fort Pond. And Wainscott Pond had very serious blooms. I think I'm going to review all of those right now. So here's Fort Pond. Again, we get a sample brought out each week. And you can see this Fort Pond is almost like clockwork in many places. Almost every summer is the same. The levels of blue-green algae creep up through the summer, usually peak sometime. And this year it was in August. And some other years it takes longer to get up to that peak, and the peak occurs in September. You may recall there were at least 2 years where the Mighty Montauk Triathlon the swimming event had to be moved out of Fort Pond because of blue-green algae blooms. Well, that didn't happen this year. The peak was earlier. Wainscott Pond is, the data there, I'll just say, is a little troubling. I'm going to start with 2019, just as a quick review. And in 2019, you can see an excursion that occurred in June, and then another few excursions later in the summer. And that microcystin is one of the toxins that blue-green algae make that we worry about. And you can see, a peak of about 8 micrograms per liter. It's 2019. And that was sort of similar to some of the prior years. 2020, the switch seemed to flip. It was always above the standard for blue-green algae in 2020 from the moment we started sampling to the end of the season into November. And the toxin levels were sky high in 2020. And these are, I should also mention, we also go through the effort of, we are ELAB certified by the State Health Department for microcystin measurements as well. So these are, again, legally defensible actual measurements that actually get reported to the DEC. You can see very high levels. And remember, in 2019, we peaked at 8. And so way over that in 2020. It's now to move to 2021. Skip the slide there. Once again, there's some missing samples where the ND is no data, not detected. But what you can see is that, again, every measurement we made was over the threshold for the levels of blue-green algae, with some very high levels measured. All those italicized words there, microcystis, Aphanizomenon, Planktothrix, Telgospermum, Anabana, these are all different genera of blue-green algae detected in Wainscott Pond in 2021. And the toxin levels were not as high as 2020, but again, much, much higher than we saw in 2019 and years prior. So a little bit better on the toxin front, but still, the standard for the EPA standard is, it says 10, it's about right, it's closer to 8. And then the shoreline threshold, the DEC calls high toxins. The DEC, I should say, should be mentioned before, but that, again, that DEC bloom threshold, at that level above the 25 micrograms per liter, they say a water body should be closed for recreational use. And then they say it's not only closed, but it has high toxins anytime it's above 20 micrograms per liter or parts per billion, which is shown here. And so I just put this up here, for a while I've been, had the idea of, we did, as you may recall, we did a very comprehensive study of Wainscott Pond and determined that the sediments in this very shallow water body are the strongest source of phosphorus, a significant source of nitrogen, and that removing some of those muddy sediments might actually remove both of those nutrient sources, as well as remove some of the blue-green algae that actually overwinter in those sediments. And unlike many other water bodies, there's a lot of open space around Wainscott Pond to actually put the sediments once they be dredged out. So I think that's something that should be considered, but there sort of needs to be some organization towards that.
You notice, I think it was 2019, the very high levels were towards September, October.
Mm-hmm.
And then in 21, I think it was, or maybe it was 20, it more or less looked like Fort Pond, where it peaked sort of in August, almost with the temperature.
Yeah. Well, the interesting thing about Wainscott Pond is that the blooms are getting more intense, and we're seeing more diversity as per what I showed here. Many different types of blue-green algae present as well. And it's just worth mentioning, some, but not all of the, 3 of the 4 generalists there make microcystin. And so the amount of microcystin may or may not be concordant with the amount of blue-green algae there, because if you have a species that makes a different type of toxin dominant, you may not get the same levels of microcystin. So those very high levels in 2020 were probably associated with microcystis being the dominant genera. We saw a little bit more diversity in 2021. Okay, and then moving on to Georgica Pond, just as a quick update and reminder, the weed harvester was reenacted in 2021 with DEC permits for the month of most of August and the first week or so of September. Over 50,000 pounds of aquatic macrophytes were removed, and this would represent up to 10% of the nitrogen and phosphorus load during the summer. There were 2 openings of the cut in the fall in Georgia Pond. So what you can see is, just as context, the salinity was quite low all summer long, less than 5 from, as you can see, from May almost all the way till October. And you see how quickly the system responds with regards to salinity. The first opening only lasted 4 days, and then it was open again, and I think then closed again thereafter. I mentioned the high levels of blue-green algae. Here's a time course of that. The highest levels were seen in the south. The salinity is actually almost 0 when you get to the north, and whatever the blue-green algae are, the genera that establish themselves in Georgia Pond, they don't like the ocean salinity, but they also don't like fully fresh water, or maybe they're getting flushed out of that particular region. But they never violated the threshold in the north of the pond, but we did see both in Georgia Cove and in the pond proper levels that were in some cases 4-fold or more above the state standard. And as per the same result we saw in 2014 and 2015, the opening of the cut ended the blue-green algae bloom. And so that's a dual response. On the one hand, you're tidally flushing out the blue-green algae, but on the other hand, they're not really well adapted to high salinity. So they can handle maybe 10, maybe 15, but you can see here the salinity pops all the way up to above 25 in the lower part of the pond. So opening the cut is a viable and successful mitigation strategy for mitigating blue-green algae blooms.
We think that, the way the dredging has been progressing, it's doing a much better job of affecting all areas of the pond.
Yeah.
Every year we try and get it a little bit better.
Yeah, and I think in the event that things, that as you can see, the blooms peaked in late summer here, but I think in the event that the salinity was low all summer again, and the blooms got very dense earlier in the summer, I think depending on what's happening with piping plovers and otherwise, there could be considerations of opening the pond on an emergency basis. That this is a human health issue, and I know the town of Southampton considered the same thing last summer as they sought to get emergency.
We're talking with Southampton.
Good.
Going to the Fish and Wildlife Service to do some biological assessments to, in hopes that we can get a incidental take permit to open the pond on an emergency basis.
Yeah, I think that's a good idea, and I think everybody wants to see the plovers thrive, but nobody wants to see dogs die or people get sick as well. So, and good news on the toxin front in Georgia Pond, when we make measurement whenever there's a DEC exceedance. We did measure microcystin above our detection limits in early August, but not later on. And the level at 5 micrograms per liter was below the EPA threshold. Final thing, we did look carefully at the opening of the cut in the first opening that happened last fall with the high cat. Already talked about the high cat, so we can just go to the images here. And so this is probably the most useful of the high cat data that we got, in that we ran a survey. I think it was actually maybe the day of the opening, but before tidal flushing had been instituted, and then ran it again, as you can see, just a few days later. And so you can see the very strong jump in salinity, with the highest salinity being, as it would be expected, right down by where the inlet is, and a little bit less over to the east, so suggesting maybe that you're getting stronger flushing on the west side with the cut open. There's that disappearance of the blue-green algae bloom. So the high levels that were present when the cut was made open, and then just a few days later, them being pretty much eradicated by both the higher salinity and the tidal flushing. And as we've always seen in the past, the oxygen being dragged down with the opening of the cut. And we have some hints as to why that is, but thankfully, again, it was not nearly as extreme in 2021. Okay, and then very last thing, just a very quick update. As you know, we've been growing oysters in Georgia Pond in an experimental setting. And so we've had now, as you can see, 3 years of oysters growing and surviving in Georgia Pond, except in the north. We lost our oysters in the far north They've been out there for 2 years, and we think it's they just got almost fresh in the far north last year. So we're continuing our oyster work, but we're sort of setting a line in the sand at Burnt Point. And so we're going to focus our efforts south of Burnt Point. And it's also sediment-wise much better there. It's very muddy up there, which no matter what you're doing with oysters, not a great oyster habitat. But we have some, you can see in the region south of there, they've done very well. By and large, we're putting out small oysters. So when you, the fact that the ones that are, these survival rates of 60% or 50% are actually pretty good considering the small sizes that we're starting with. We also did an interesting experiment last year where we put out oysters in 2 different ways, pre-acclimating them to low salinity and not doing so. Our oysters are spawned at a high salinity. Most waters, of course, across all of Long Island are at a high salinity. And so what we found is that by pre-acclimating the oysters in this case, which is what we call spat on shell, so the oysters growing on shell, I'm sure you've all heard that from Cornell and others, that those oysters did very well. And we had near complete survival of those oysters when they were grown, just give it a few weeks to adjust to a lower salinity, instead of just putting them out from, say, a salinity of 30 and then suddenly dropping them into 5 or lower. So obviously that's an approach that we'll continue to use going forward. And I will say this is because our first round that we did during the summer, we didn't acclimate them. And to our surprise, the spat on shell did very poorly, very low survival. So we're able to figure that out. And you can see this was a fall experiment, but it was very telling.
Why does that north acclimated seem to go to over 100% survival?
Yeah, the way we do this is we have the spat on shell, and so you're literally just counting them on there. And so, probably should just be 100%, but it probably the counts, they counted more than they started with. It could have been a natural set, but although given the time of year. Okay, so with that, I'm done. These are the highlights. I don't know if I necessarily need to read through all of them, but I think there's a lot of really good information that we learned this year that tells us about the function of these systems, areas that need attention, some obvious, in my mind, follow-up work to be done, and also probably some good connections to good remedial water quality projects that could be taken on as well. So yeah, and so with that, I thank you. It's definitely thank the trustees. It's been an extremely rewarding collaboration. Almost a decade of data. So I think we're building a nice data set, and I've learned a tremendous amount. And so
it seems that the longer chain of data you get, more areas it points you at.
Yeah, and
it's valuable.
Yeah, and it's allowing us to pick up longer term trends, have more confidence about, when you see something just once, well, maybe it's a fluke, but if you see it year after year, you know there's something to be followed up on.
Thank you.
Thanks for hanging in there. I know that was probably very long.
No, very thorough. So I will give you a call and we'll set up our next meeting with our subcommittee then.
Wonderful.
So thank you so much.
My pleasure. And I'm going to stop sharing. Is that a good idea? Very good. Thank you.
Yeah, thank you. Give me some ideas. Can you send me this Nappy Harbor presentation? And then after that, I'll look at it. I'll call you. Hey, it was very thorough, terrific report.
Incredibly,
Right?
Who's working at it?
Yeah, we have a 75-page report from Dr. Goldberg, so good weekend reading.
Yeah.
Thank you
John,
for
did
coming.
you want to talk about the piping clover?
Yeah, I just thought I'd sort of go over the issue with clover monitoring for the board and for the public as well. Actually, Natural Resources has been, these are all old clover work and trustee territory anyway is being done on coastal beaches, and it's being done basically has been done for many years by the Natural Resources Department. And they're having trouble this year finding monitors. So I just kind of wanted to go through the process, what it entails, and if anybody's interested in participating in any way, this would give them an idea of what that means. So first of all, there's a training session by the U.S. Fish and Wildlife Service by Zoom in there's one on April 26th. The same one will be given again on May 24th, which would be helpful for someone to attend to get the idea, get the rundown of what monitoring entails.
Can we put the link on our website?
I have posted this information from about the piping clover program to social media, and I believe it's on the website. I'll take a peek. I'm pretty much every day on it to update, but I have been following along, and so I will double check that.
That'd be helpful.
Yeah.
So on trustee territory, the sites that have hosted nesting pairs of birds have been basically Wainscott Pond, Georgia Pond, Main Beach, 2 Mile Hollow Beach, Sammy's Beach, the beach at Maidstone Park, and the beach at Louse Point. Samantha Klein in Natural Resources is the point person on plover monitoring. She would train any monitor and interact with them through the season.
What is the season?
The season is basically, nesting begins in late April, early May. So sometime in that time frame, say May 1st or so, would be the beginning of any monitoring. Sam identifies the nests and then puts the nest locations on a map for the monitors. A monitor can work with one or more sites. The first task is basically to monitor the nests with the eggs in them. They're breeding, they're laying eggs. That lasts about a month. That's relatively simple. That would take about maybe a half hour per site. You're just making sure the nest is still there, the eggs are still in it, and going on down the line. Come late May, early June is the hatch, basically. And that's probably the most critical time for about, I believe, a month after, let's say, between June and July, when the chicks are hatched, they're running around, they're being fed, they're looking for food, but they're not yet flying. So that's their most vulnerable time. It's also the time at which the chicks are kind of hardest to monitor, because they're running around, they're hiding. You might have to spend a little more time. So she said up to a maximum of maybe 2 hours per site, because you might have to sit there and just wait for a chick to show itself so that you can make sure it's checked off as still being around. And then I believe it's, let's see. Once the chicks begin to fly, they no longer need monitoring. So that probably puts it sometime into July. Now, there's a caveat here in that nests can be lost by storm or predation or some other reason, and then the parents would reestablish a nest. So nests may continue to be made into June or possibly even into July. So there may be new nests through this period. And those would have to continue to be monitored and the chicks monitored after that. So basically the whole process theoretically goes through through August. So the visits to the sites are made once a week. And as I said, a monitor can monitor one or more sites, depending on their availability. And the monitor can pick the day of the week. You kind of look for a nice day. You don't go in the rain. You don't want to go in the heat of the day necessarily, or when there's a bazillion people on the beach. So, you might, especially when the chicks are running around, you might want to pick early morning or early in the day or later in the day when it's not quite so hot and the chicks might be more active. But there's flexibility in terms of, day of the week and, when you have to go. It's not locked into, Monday morning at 7 o'clock like the mosquito. There is community service available for this activity, and there's also a stipend available for a monitor. We talked a little bit about Georgia cut and the possibility of moving towards opening the cut out of clover season if we can get a permit from the U.S. Fish and Wildlife Service. So I thought this might be of particular interest to us in light of our efforts to move forward with a permit from U.S. Fish and Wildlife Service in conjunction with Southampton Town to get the emergency opening. If we have a hand in plover monitoring, that might actually help with that, getting that permit from Fish and Wildlife Service. The same
Plovers goes a long way with what they let you do.
The incidental take permit.
So that's the rundown basically. So
Good.
for us to think about, for hopefully somebody listening, they should contact Natural Resources directly or our office, I think Natural Resources Department
Did the Department of Natural Resources or the town of East Hampton issue a press release on this?
I don't know.
Okay. I'll call Sam.
I know they've talked to some people that, there is some interest from some people that have worked, for the town in the past.
Okay. I will follow up certainly, but I know I did put it onto Twitter and our Facebook page. I'm not sure I got it onto the web yet, but I will see tonight.
Yeah, and also for the community service with the schools,
They get credit.
the high school students need community service, and if you're getting into something with nature or marine biology.
You know what, a good organization is through Aubrey Peterson with the East Hampton Environmental Group, his after school program.
I did talk about high school students and community service with Sam and Kim. And there is a little bit of they're a little young, they don't drive necessarily, there may be, if they're paired with somebody who, does drive.
And they disappear in July and August.
Oh, that's could be true.
Well, this could coincide well with a college student who might come back from college in June and then go back to school in August. They would be here for basically the period of the monitoring. So that might be something that in conjunction with another job they might have at night, there's money available. We had several college students on the mosquito larvae.
I was just gonna say, this
Yeah.
sounds exactly like the situation that we have with the aquaponic salt marsh research program. Same thing,
So,
finding the students. But certainly ones who are interested in wildlife and marine biology, that's where I had always stopped in the past. First year of school. All right.
Do you have information on kayak rack? Or?
So we had those kayak rack signs, and I found with just a regular drill, I could have those centered holes.
Oh, okay.
So then that's why I was able to just put them up on Louse Point, where there's 3 trails go down to the rack. So I think those are pretty good spot for those. And then there was one at Mile Hill over in Northwest Harbor, a big one that says the same thing. So I'm like, let's leave that. And then, so we'll just chip away and get the rest of them in this week.
Okay, great.
So what's left to do?
Settlers Landing that's 6 racks there that I was going to do, but I don't have a drill. I have one that needs an electrical cord, so.
All right.
Settlers Landing,
Batteries these days.
Fresh Pond, was it still, Arlene?
Yes, it's just one rack there.
1 rack or
Fresh
2? One or 2?
Either way, I'll just bring extra signs, so.
All right. If you'd like, do you need some help? I mean, I can come along and help.
Yeah, I mean, I'll let you know if I can't figure it out. So that's the only 2 we have left, so.
No, we have Settlers Landing, Fresh Pond, and Gerard Drive.
Oh, right.
any kayaks on the racks yet?
No.
They all know that they're not supposed to go on until they have permit stickers on them.
Yeah, so far so good.
So Mile Hill, though, even though there's that one sign there, you still should put them on the racks
Okay.
themselves when people actually walk up. I mean, last year I had a fellow,
That's right on.
Yeah, the sign is on the rack.
right, but it's sideways, so when they actually walk around to put their kayaks up,
Okay.
I think we made them for there.
Yeah.
So I think they should be there, because I had a fellow last year right at Settlers Landing. I was nailing them up and the signs were all there. And he stopped his car and he said, "Do I need a permit for this?" You know, it's the sign.
Sometimes people have a story too.
Yeah. Yep, all right, great.
Yeah.
So,
And then moorings is next.
That's it.
yep.
Well, yeah, I think there's not really much to moorings. I understand there are only 8 more spaces left in the large boat grid. So if anybody has a large boat and wants to moor,
Better get on it, yeah.
call the office quickly. I think that's the only thing.
And the number is,
Yeah, that's pretty much it.
and the number to call
Are we
is,
gonna,
631-267-8688.
that's right.
we're gonna stake out those spots?
They're staked out.
No, they're done yet.
Looks good.
Babes Lane and Louse Point. The southern end of Louse Point is staked out. I think we've spoken to almost everybody that there was some question of whether we should be doing it in Northwest Creek. But we've spoken to almost everybody in that vicinity and they seem to be clear on.
Yeah, they've been doing it, it's like a little community down there, doing the same thing for a long time. So Susan, do you want to talk about the scholarships?
Sure.
Balloon band?
Because I thought about it yesterday, and sure enough, today I received an email from Stephanie from the East Hampton High School, Stephanie Otto, that our application, our package
A
is
group of people.
ready to be picked up.
They're all,
Yep, and so I will bring those back to the office.
you have to judge them?
Yes, it's the same as last year. We do have to discuss what we're going to do this year in terms of 1 award or 2, as we have been doing in the last 2 years. So I'm always in favor of 2. You know, we're trying to grow our scholarship fund, and today, I'll go to this, but you know, if you are cold and you want a sweatshirt, this adds to our fund when you purchase
It's
one
staying
tomorrow.
warm and help somebody go to school.
Exactly, stay warm, yep. So the Captain William J. Ryson Scholarship Fund is our fund, as you all know, for children, students who are graduating from East Hampton High School and who are going on, and this helps them with their school tuition. So the applications we received from the East Hampton High School, I'll pick up tomorrow. And I'll bring them back to the office. They are anonymous. They're numbered, so we don't know who has written them. We read each one, and then you put in your vote. And you vote by the number on the essay. And I really encourage everybody to please come down and read them. This year we tailored it to be more specific to trustees, so that it was really someone who's interested in what the trustees do. They understand what the trustees are, or history. And we have asked them in the past to provide us with an art piece or a poem or a painting or something, but that is definitely directed to the trustees, and they have an understanding of who we are and what we do. So it's much more specific this year. So I am hopeful that we will, and I guess we'll discuss this on the committee, education committee. David, you're now on the committee.
Why don't you make a motion to give 2 scholarships?
We have a quorum, but why don't we?
What's the timeline on that?
Yeah, why don't we wait
Okay.
until we have a full board?
Yeah,
I think that would be best when
Yes.
we have, because we're missing Jim this evening and Francis and Mike and Ben. So I'd like to have a full board. We've done it in the past, and I think that we
Yeah, it's
will probably
Probably a short
agree to
thing
that.
along, but-
I mean, maybe, I don't know. I'd like to know more about the balance between giving 2 scholarships and building the fund, which we started off with. You know?
Right, well, that has been, the actual building of one it requires, we had talked about investing. And we asked Ben to look into an investment plan through his agency. He was going to come up with some information. When Rick was here, he was going to come up with some information as well. So yes, the idea is to grow the fund so that we are actually giving what we give to a student or two is from the interest derived and earned from that principal. And that has been a,
Yeah, but the fund was not in the healthiest shape, so,
Yeah, I heard.
yeah, so we're in a building process also.
yes.
So you got a balance between,
Yeah.
you know, giving it away.
Mhm. We have in the past had an anonymous donor deliver the second award, specific to that second award, that donation. But anyway, I'll pick up the essays tomorrow from the school and bring them back to the office.
And
Susan,
again, it might
if I
be,
can jump
it
in.
might be more
Sure.
interesting to have the discussion after people have had a chance to look at the application.
Yes, sure.
Yeah.
Arlene had,
Yes, when Susan joined the board, we had $24,000 in the Wrestling Fund, and with Susan spearheading it and Jim Grimes supporting her, was it 3-4 years later, we have 60,000 from 27,000. We are designating 10,000 from our dredging receipts from Georgia Pond a year into it, and of course the t-shirts and all of that, and anyone that donates, anything that's donated or generated, largest clam contest or anything, everything goes into the scholarship. So 24,000 to 60,000 in
That's
3
great.
or 4 years is yes.
we're moving along. All right, so that's that. And
Have any international news about the balloon ban?
yes, international news. Yes, Mr. Walsh was spotted sent me a picture. Thank you very much, Chris. I appreciate that. And he said it's traveling globally, and he is there in Bangkok wearing his Save a Fish and Balloon Pollution t-shirt. So thank you so much for that. And Mr. Walsh is sitting over here in our audience. I quickly put this in because I was asked for May 8th to address the Ladies Village Improvement Society about balloons. And since LVIS is in the business of a charitable organization to beautify the village of East Hampton, I was delighted when I got a call to ask me if I would do this on May 8th, and David was going to be of help. We were going to present at the Coast Guard station. But it got switched, and suddenly yesterday I got a call from the woman who puts together the programs, and she said, I need you tomorrow. So that's why I'm wearing my sweatshirt, because I did a presentation today slide presentation of only pictures of balloons seen in East Hampton town. And you were in my slide presentation with all your balloons when you did a beach cleanup in Montauk. And Mike Martinson, our trustee, also with balloons. So I think it was a nice program. I hope Gail enjoyed it. I mentioned you, as you know, I try to name everybody I can. Sorry, Chris. But so that was fun, and it was good. And I asked the ladies if they would support our efforts, my efforts, to see if we can get a ban on the sale and distribution of helium and gas-filled lighter than air balloons in the village of East Hampton, because after all, Ladies Village Improvement Society is beautifying our village. So let's keep it beautiful and clean. And they seem to be very receptive, and we're applauding. So I hope that I will move forward now and ask to have a meeting with the village town board, the village board, and the mayor. I've already put in the call, and I'm waiting. I'm waiting to hear. I will wait a little longer, and then I will pounce. No. No, but it's a good thing, and I think it's time.
You have a powerful ally in the LVIS.
Yes, it could not have happened at a more opportune time, because they came to me and I said, bingo, if the Ladies Village Improvement Society, of which I am a member, with Gail Aldred, John's wife, and among other people, if they are interested in seeing this, then they will be a good sport.
They're powerful force.
Yeah, it's a great organization.
Yeah, they beautify, all the flowers, all the trees, everything that they do. Yeah, and there's nothing worse than seeing a balloon wrapped around those beautiful flowers and plants. So I'm hoping that we can move along and get that done. I'm waiting. I did have a webinar class that I attended with a group called Beyond Plastics the other night. And Assemblyman Steve Engelbright was the guest speaker, and we actually were able to accomplish what we were trying to do, to a vote to not pass a bill that would have been actually putting us back. It was about plastic bottles, and that's something that we have been working on in Suffolk County Single-Use Plastic Task Force with legislator Carl Hahn, and that has kind of fallen a little bit by the wayside, which is why Courtney and Surfrider, it's now become a regional thing where we are trying to get restaurants and places where you take out food to not give you automatically plastic forks and spoons and knives, but rather ask the customer, are you going home? Because if you're going home, you have your own ketchup and your own mustard, and you don't need the condiments, and you don't need utensils.
Tim, what's this slogan?
It's
save the stuff. No.
Some stuff is.
Yeah, it was something. Skip the stuff.
Skip the stuff, yes. It started out over a year and a half ago now with the Single-Use Plastic Task Force, but because it's an election year, things have kind of switched out to a regional general thing. So we're going to start this process, and I did write to Courtney and say, yes, I'm on board, because I've been working on that. And we actually did posters and everything for restaurants, and it's just kind of fallen away. But that's our next effort, to keep our environment.
With the pandemic and everyone doing takeout, it just became,
It became more than that. And
we went way backwards. Yeah. And so it's just a good start again.
I was actually at a lunch not too long ago in the village, and they are giving plastic straws out. And that has been banned. So I'll have to tiptoe in and say something nicely.
Put that on your meeting agenda with the mayor.
I will. You laugh. Listen, little by little, my square wheels of justice move forward. Sometimes they move backward.
That's the whole thing with any of this stuff. You just got to keep pushing.
I'm persistent.
Incremental progress.
Yeah, I'm persistent. I'll take a little bit at a time, slow is the turtle, but to the other side of the road I shall go.
Great.
And that's it for me.
Thank you very much.
Before we move on, Susan, I wanted to add a little bit about the William J. Rice Scholarship Fund.
Yes.
So Susan had asked me several weeks ago to begin to look into whether or not this fund would be appropriate for the 501c3 tax exempt status. So I began to look into that. I did speak with our accountants as well. a tough time of year to bring them in on those types of
Yeah,
things.
taxes.
But ultimately, it is a bit of an exhaustive, sort of comprehensive application process with the IRS. It does seem like on its face that this might be the type of charitable institution that would, be eligible for and benefit from that. So certainly the door is open there, but we're going to keep looking at that application. I'm going to speak with our auditors as well with the town. They might be able to help out as well, and then sort of continue to review the application and hopefully bring on somebody from the accountant side of things to sort of help us through the application process to see if it's going to be the right fit for us. So while I can help the trustees legally, I'm not confident that I have the CPA skills to guide us through this entire application. So I'm going to keep an eye on that in the next couple of months and see if that's going to work out for us as well.
Great, that's terrific. As I told you, I went through that process with LTV when they became a 501(c), and it took a while to do, but it's definitely worth us trying to do it. So will we ask our own accountant to assist us in this?
So yeah, I asked her originally what her thoughts were. She said she's dealt with cell before, but it's not her expertise, and just that kind of these couple of weeks should be tough for her. But we'll revisit next month.
Okay.
She did mention speaking with our auditor as well for some guidance.
Great.
So I'm just going to keep plugging along, and I'll give you updates as we go, and hopefully we can get this application there in the next couple of months.
Excellent, excellent. Thank you so much for bringing it up.
What's the advantage to that over whatever the status is now?
Taxes, when we sell things, we don't charge taxes, number one.
This would be
Just
primarily for the sale of these goods.
Yes, or anything that we do with this particular, with the Captain Rysam Scholarship Fund. Anything we sell, there's no tax. And anything that we buy for instance, if I do a fair and we need to buy things, I now would have a 501(c). We would have a 501(c), and we don't have to, we're not charged taxes. It's a very good thing because it's not for profit.
If people made donations, that would be a tax.
Yes, we're not making a profit on any of this. So rather than charge taxes, which is a little more complicated, it's a good thing for us. And people like to be able to write off donations. And if we're a charity charitable organization, that would be very helpful to us. And it gives us more credibility as well.
So see what the process is.
Yep, relying on our Esquire over there. Okay, thank you so much for that. I appreciate it.
Yeah, thank you very much. We just have two bills for the administrators. Patrick Bistrian for doing our annual maintenance on the culvert, and against the post office box. The Bistrian bill is $29,675. Against the post office is $332. Can I get a motion? I'll make a motion, we pay the bill.
Second. Third.
All in favor?
Aye.
Any opposed? Passed and carried. I haven't had a chance to look at any of the financial revenue. Under the weather, with most of the world,
No.
Want to stabilize? Let's do it.
Yes, please.
Okay. Do you want to? You're going to talk about the real estate thing. Oh, we've had a complaint about the area of the dock. We're going to look into it. The Bell Estate. It seems like the area we're talking about is private property. There was a time way back when the trustees sold their rights to the beach or traded their rights to the beach in exchange for the fresh pond itself and all that parkland. So I think we should contact the code enforcement, have them look into it. There's also the town had a similar situation a couple of years ago with a piece of distressed bulkhead between the lifeguard beach at Little Alberts and the truck beach down there. And they wound up removing it. So we have to identify who the person would be that would have to do that and maybe find out what was involved. The photographs are, they're they're ugly. Yeah, they show a bad situation.
Photographs show erosion in an old bulkhead field north of Albert's Landing Beach.
I mean, those bulkheads have been there for 100.
And that
And
seems
the
to be,
groins, the two
that
groins.
seems to be the southern, that bulkhead that's exposed now seems to be the southern extent of
Yes,
the bell switch.
yeah,
Trustees.
there are no trespassing signs down there and, other things. So if you want to just look into it a little and see if we can,
Francis sent us all an email. I had actually received a phone call over the weekend from the resident of East Hampton who had described what was going on, and it kind of grew, with the pictures. And she's been walking up and down the beach, and
she's very, very knowledgeable, and she's been doing it for a long time. She was involved, I think, in a last town, and the town, removed that part of the bulkhead. It was on property that they own,
right.
we own. So, yeah, I think identifying who owns it and seeing if they got permits to put that fill there is.
Well, I'm told during the telephone conversation that this is a new purchaser who's now been doing some things, and a new purchaser is going to be building and changing that property. If you look at a Google map, it's the property is at the end of Little Albert's Landing, and there's a path down to the house, and it's situated between the two lines.
Yeah. There's permitting processes for all that stuff.
Right.
Natural resource, there's a lot of stuff, so you just want to make sure everything is being done the proper way.
way. So, okay.
So how's it feel to be back
Good.
in this building?
I know, it's been, what, 2.5 years?
There've been some, it looks like really improvements with the camera systems.
Yes,
Yeah?
new, and this is, our speaker is right here. We have one
So
little
I'm
speaker.
hoping
Microphone.
that this little,
That's a microphone.
What did I say, speaker?
yeah,
Oh, sorry. Camera.
My first meeting, I had to wear shoes at.
really? You can't get up and casually walk.
Yeah, no pajama bottoms.
We want to be there.
Yes.
But there is, it is nice to have the interaction.
Yeah, it might be a little break after this one.
Yeah, but, everybody's wearing, a lot of people are wearing masks because, there seems to be a little bit of a milder version of this around. We just recommend that everybody not declare victory too soon.
That's
Yeah.
true. Well, there is a rise in New York City. The mayor has now mandated that all students
Mayor has it.
wear masks to school. That's a new mandate. And Philadelphia today announced that any closed buildings that people go into, they're the first city to do this. You must wear a mask inside. So we'll see where we're going. But the thing is, now with at-home tests, people are not reporting that they have COVID or the BS, whatever it is, BA.
Yeah.
So we don't have legitimate numbers. We don't
Yeah,
really
no,
know what's going on, but it is milder for those of us who are vaccinated.
There is a way you can use the home test and register it. There's a website you can actually go on, actually like film you have to keep the website going for the 15 or 20 minutes that you do it, but
How many people are going to do that?
yeah, well, people would need it for travel or something.
That's
like that.
true.
Might do something like
Well,
that.
one of my grandson's preschool class had a student that tested positive, and the class has to be out for 10 days and take these tests and, prove that they tested, not prove, but they're trusting them, testing it negative before they come back, so.
Honor system.
So anyhow, we'll
We'll play fair.
play it by ear. Hopefully in 2 weeks, we'll be back here, all of the smiling faces. Hanging out.
Well, I'm going to continue wearing my 2 masks.
I have a lot of masks.
Good. Thank you, everybody.
Thank you, Bill.
Motion to close?
Second.
Motion, oh, second, who?
All in favor?
Aye.
Aye. Okay,
Thank you
good
very
night.
much.
Good night, everybody. Thank you for coming, Mr. Walsh.